All right, welcome back. You are now in the last little bit, just 3 more weeks, and then you never have to see me again or study for this exam again, right? It's been a pleasure. Many of you guys will start to disappear probably. Many of you are getting sick of school. I get it. I'm sick of it too. But we got to finish up, talk about gram-positive bacteria. At the end of this section, we're going to talk about archaea. Not many of your classes that you had today really delve into some archaeal discussions. We will do that. And then we will finish the semester with 2 more papers, just like we discussed with BLPG. Those will go online by Friday. Also on Friday, you will get your second to last homework assignment, and you'll have a week to do it, 1 more to go after that. A reminder to please consider any extra credit assignments that you would like to do. The semester will wind up quickly. Questions, comments, and/or concerns? Perfect. So, if you'll remember, we left off discussing the actinomycetes, and specifically we left off on streptomyces. So this is the last actinomycete, or the high G+C organism that we'll discuss. So streptomyces, again, a reminder, has large linear genomes. They make up a significant portion of the total soil population. These really massive genomes have a lot of metabolic diversity. That is, they have a lot of different transporters to take up really weird compounds in the environment, and because of that, they can make a lot of really neat secondary metabolites, including those that are clinically relevant. One thing I wanted to point out, especially since we just had our phage exam, streptomyces has a built-in phage resistance system, which is kind of neat. So this is phage specific. There is a streptomyces bacteriophage called Phi-C31. And this particular system provides resistance, so this genetic system provides resistance to Phi-C31 infection. So if we look at panel A, we see streptomyces growing on a plate, and we try to infect it with this particular bacteriophage, and nothing happens. Okay, this is not all turbid plaques or anything like that. There's just no infection here. However, researchers were able to isolate mutations in this cell line, this so-called PGL- strain. Now all of a sudden, this bacterium becomes susceptible to this particular phage. So by doing the genetics, the researchers identified 4 distinct genes and proteins that are required for this resistance. These genes modify DNA and protein through methylations and phosphorylations. These modifications basically shut down the phage's ability to continue replication. So you actually get an initial infection, the phage injects its DNA into streptomyces, and as it begins to replicate, these genes in streptomyces work together to shut down continued replication. So basically it's kind of a built-in immunity. Now, one of the other interesting things was in the observation that one of these genes, in this case PGLX, that's involved in this phage resistance system, has this stretch of G residues. Okay, it has a repeating stretch of 8 Gs. And this is the wild type situation. That is, this M145 locus sequence comes from this strain that provides resistance to phage infection. Interestingly, the isolate that is sensitive to phage infection
on the right-hand side here, you sequence PGLX, you actually get an extra G in there, which causes this PGLX protein to shift out of frame. However, different isolates of Streptomyces, in this case you see this J1902, at that PGLX locus, it has only 7 Gs, and that's sort of the normal situation. So it looks like this locus at PGLX can sort of expand and retract in this sort of repeating G section. And it's thought that this modification or this expansion or retraction of that G tract might provide resistance to different phages in the environment. These are so-called phase variations, that some strains express a certain PGLX, other strains express a different PGLX, which may have other functions. Okay, so we're going to wrap up the actinomycetes. I showed you that they have fungal-like growth patterns. They differentiate, they form aerial and substrate mycelia. We talked about natural soil decomposers. They have extensive secondary metabolism. And again, we talked about mycobacteria as a model pathogen and actinomycete. Okay. Below G plus C are the firmicutes. And these are the more typical bacteria, if you can imagine such a thing, standard rod or cocci-shaped bacteria, including Bacillus and Lactobacillus. However, even though they're typical bacteria, we're going to start with something a little bit bizarre. And these are the mycoplasmas. And these are the class molecules. Do not confuse mycoplasmas with mycobacterium. Mycoplasmas are so-called plasmas because they actually lack a cell wall. And they have highly reduced genomes. It's thought that these arose through some ancestral gram-positive that went through a genomic reduction. And because of this genomic reduction, they always have to be associated with some host, although the host range is quite diverse. They lack the ability to make certain peptidoglycan precursors and other standard metabolites, again showing that they have to be associated with some host. So this slide shows you the size of the genomes we're talking about. We're talking about this highly reduced genome. So for example, this mycoplasma genome can be as low as 580 kilobases. So this is a very small genome. And we find the mycoplasmas or ureaplasmas associated with humans and other animals. However, you can see the diversity in the host patterns here. Some of them are associated with insects or plants, right? But they must be associated with the host. Because they're a because they're a plasma that lack a cell wall, they can take on many different shapes, which we call pleomorphic. And here's sort of an example of the different types of shapes the organism can take. So you have these sort of extended bacterial look. We got some that kind of look like pear-shaped. And these organisms tend to have special structures on them called terminal organelles. And this is going to be really important for the organism's motility as well as their ability to associate with a host. And because these organisms lack a cell wall, one of the critical things they must take from their host is a host sterol. For those that are associated with mammalian hosts, that's cholesterol. And we take host cholesterol and we put it into our membrane, which allows these organisms to be stable. And in mammalian systems, these organisms tend to be mucous membrane pathogens. So this is very often the cause of like walking pneumonia you might have. You have a cough that lingers for 3 or 4 weeks and doesn't clear up. Well, very often that is mycoplasma. One of the genera within the mollicutes is the so-called ureaplasma. Ureaplasma is a normal urinary tract flora member. It's possible that can cause infections. However, this organism is interesting due to the ability to utilize urea to generate a proton motive force. So in this organism, it has a special system for producing a urease, which is the enzyme responsible for degrading urea. This is a nickel-dependent enzyme. We convert urea to carbon dioxide and ammonia, and we can export that ammonia where it will release a proton to generate a proton motive force that we can then bring in down an ATPase to generate ATP. Mycoplasmas are also often studied for their ability to have a bizarre gliding motility. For example, Mycoplasma mobile is an organism that produces this so-called terminal organelle, which is this sort of the smaller end here. And these organisms appear to have a cytoskeletal extension. So these are extensions that come from inside the cell to contact a solid surface. And during ATP hydrolysis, these attachments sweep back and forth. And when they're associated with the solid surface, they can actually pull the organism across the surface. Similarly, in Mycoplasma pneumoniae, you have this terminal organelle that can kind of extend and retract, much like a type 4 pilus that we discussed, with these cytoskeletal attachments that are associated with the surface. So this thing is basically almost like a snail moving across the surface. Again, all of these motilities are ATP dependent and require cytoskeletal extensions. Okay, the class Clostridia. This class is polyphyletic, which means that these organisms are actually quite diverse at the genetic level, but they've all developed similar traits and phenotypes that have evolved independently from unrelated organisms.
Why are they put in the same class then? Like,
Because,
they're
right,
similar.
absolutely. So why do they do this? Because they started making these trees a long time ago without significant genetic information. And they have kept the Clostridia together, even though it's pretty clear that at the genetic level they are quite diverse. This might change in the future. Clostridia are strict anaerobes. Oxygen will kill them. And they are one of the 2 classes that form endospores. Some of the genera, within Clostridia, actually stain gram-negative, which is why you should never completely trust your gram stains, and why we take them down to sort of the genetic level now. So here's a micrograph of our favorite Clostridia. This is Clostridia botulinum. One of the common features from Clostridia is their ability to ferment amino acids. And in order to do this, they must both oxidize, which is the left arm, and reduce, on the right arm, amino acids. So the end product for both of the oxidation and reduction is acetate. And you can see that each one of these arms allows us to produce ATP. And we have to couple the oxidation and reduction together due to the fact that one arm produces the reducing power in blue that will eventually feed into the reducing arm. We also, from this pathway, can generate ammonia. And this is the common source of that Clostridia smell. So when you're working with Clostridia in a laboratory, you know when you're growing Clostridia. This is the so-called Stickland reaction that is using amino acids in both the oxidation and the reduction pathway to produce ATP. Now, Clostridium botulinum is obviously an important organism. Not only is the entire food industry revolve around getting rid of Clostridium in the food source, right? In the food chain. It's also critical for, staying beautiful forever. And the reason that this works is that this potent neurotoxin that's produced by Clostridia has a very precise activity in cleaving these highly specialized snare proteins, which are critical for driving neurotransmission. So as the synaptic vesicle that contains a neurotransmitter, in this case acetylcholine, approaches synaptic cleft, these snare proteins must come together to form a complex to drive the fusion of these vesicles and be able to dump neurotransmitter. These botulinum neurotoxins can cleave all of these different snare proteins. And when that occurs, neurotransmission is halted, and then if you take enough, for example, if you ingest up to 90 nanograms, which isn't very much, you make a dead person. So Clostridium tetani, which is the causative agent of tetanus, right, actually utilizes a sodium motive force as opposed to a proton motive force to drive energy generation. If you look at the genome of this organism and you predict the proteins that are produced, you can see a high reliance on sodium, either symporters or antiporters. For example, carbon source and amino acid uptake is completely powered by sodium symporters. And therefore, this organism must utilize proteins to pump sodium out of the cell to maintain the sodium motive force. To do this, the organism has a number of sodium ion exchangers, so we can move sodium out of the cell in exchange for protons. We have a large number of ATP-powered or pyrophosphate-powered ion transporters to export sodium out of the cell. Interestingly, we have protein that you would expect in standard oxygen respiratory chains. Right? So these are an oxidoreductase. However, instead of pumping protons, it has evolved to pump sodium out of the cell. So this organism now has completely re-engineered itself to ignore a proton motive force in exchange for a sodium motive force. The other class in the gram-positive low G plus Cs that form endospores are the class Bacilli. You all know and have heard about Bacillus subtilis, which is sort of the type strain used when we discuss the bacilli. They're normally soil microbes that produce some antibiotics, for example, polymyxin or bacitracin. One interesting bacillus is Bacillus thuringiensis. This organism is often used as a treatment for the environment to kill insects. For example, when you're out spraying for mosquitoes, you're actually spraying Bacillus thuringiensis. It is not a pathogen to humans. And one of the reasons it's toxic is the fact that it produces these so-called parasporal bodies. So we're looking at this phase contrast micrograph of Bacillus thuringiensis. The spores are shown in white. And right next to these spores, you see these black bodies. These black bodies are a lattice of protein that always form in sort of next to spores. And these parasporal bodies, which are shown even sort of more clearly here in these electron micrographs, when ingested by insects, the lattice breaks down and releases this pore-forming protein that works on the insect epithelium. So you essentially poke holes in the mosquito's gut. In the laboratory, you can actually purify this parasporal body, and it's actually toxic to a lot of different cell types, but it only solubilizes in insects. Staphylococcus aureus is another pathogen that you've all heard about. They are facultatively anaerobic, that is, they can grow both in the presence or absence of oxygen, and metabolizes glucose to lactate. These tend to be skin-associated organisms. Obviously, you're all aware of MRSA and VRSA as methicillin and vancomycin-resistant organisms. But the reason that these organisms are such important human pathogens is due to their ability to accept environmental DNA. They are highly naturally transformable. Within the order Lactobacillales, we find the lactic acid bacteria. They all metabolize, or they all generate ATP by substrate level phosphorylation. However, they have a limited biosynthetic capacity. They lack the ability to make certain amino acids and vitamins. This organism has been co-opted by the food industry to make things more delicious, and it can ferment to lactate via two distinct pathways. The one shown on this slide is the so-called phospholipase or heterolactic pathway. In this pathway, we're fermenting glucose to lactate, and we produce carbon dioxide. However, there is a different pathway that exists that some lactobacilli can actually take glucose to lactate via this so-called homolactic fermentation pathway, which does not produce carbon dioxide. In the phospholipase pathway, ketolase pathway, you produce lactate, ethanol, carbon dioxide, and this organism is used to ferment things like sauerkraut, and pickles, and yogurt. It's one of these generally recognized as safe organisms. Whenever I show metabolic pathways, for example, like the phospholipase pathway, as well as the Stiglitz reaction, you have to know things that I specifically point out. For example, that the phospholipase pathway produces carbon dioxide. You do not have to memorize these pathways at all. Josh.
You said it was the phospholipase pathway that's used to ferment foods.
So actually both pathways are used to ferment foods. However, the phospholipase pathway is the only one that produces carbon dioxide. Within the lactic acid bacteria, we also find Streptococcus. Streptococcus and Staphylococcus are very often confused. Don't confuse them. Streptococcus is a lactic acid bacteria, Staph is not. All of the Streptococcus are also facultatively anaerobic. We can identify a particular Streptococcus by looking at its ability to produce a hemolysin. So we can put these organisms on blood auger plates and watch their ability to lyse those red blood cells in the media. The alpha hemolysin producing strains produce an incomplete lysis. I'll show you pictures here in a second. The beta hemolysins show complete lysis. You get a very distinct zone of clearing on these plates or on colonies. Some Streptococcus strains are so-called pyogenic, that is, they have the ability to produce pus. And these strains are almost always beta hemolysin producers. So here's an example of certain Streptococcus strains on blood auger. It turns out that there's actually gamma hemolysis, which basically shows no hemolysis, so I don't even know why they designate it as a hemolysin. However, in beta hemolysis, you can see this very clear zone of clearing on these plates where the organism has been able to completely lyse and degrade these red blood cells. Alpha hemolysis shows some sort of intermediate ability. You don't get really obvious clearing, although you do get red blood cell death. And again, gamma hemolysis, don't even worry about that. Okay, so those are all the gram-positive low G+C organisms that we need to discuss. We talked about the lactic acid bacteria, our endospore-forming bacteria, as well as the molecules, as in the context of mycoplasma and ureaplasma. All right. So the archaea is not something that we often discuss, right? And when researchers started to work on archaea, they didn't know they were working on archaea. These organisms came into the laboratory because they had some funky abilities to do some interesting chemistry. In this case, they were able to make methane. And this was probably in the 60s and 70s, and researchers were like, wait, making methane is pretty cool. And it seems to be pretty distinct among this group of organisms. But if you put these organisms under a microscope and you grow them in the laboratory, it's pretty much like growing bacteria. They look like bacteria. Same basic subcellular organization for the most part. However, the more and more researchers started looking at these organisms, they realized that there was something very different about them at the genomic level. And it was pretty hot at this time to start looking at 16S ribosomal DNA or RNA sequences. So some researchers at the University of Illinois decided to take some of these methane-producing organisms and look at their 16S ribosomal RNAs. And what they found was very distinct. And it was clear that the organisms that were able to produce methane were very different than other known bacteria. It turns out that we'll discuss this paper that came out that initially describes the archaea. I want you all to appreciate how that was determined and what the researchers went through to find these organisms were very different than bacteria. It was originally thought that these methane-producing bacteria were just very diverse, and they were acquiring the ability to produce methane from some horizontal gene transfer. They picked up the methane-producing DNA from the environment, and now they can actually produce methane. However, we would eventually find out that these organisms are all closely related to each other. As of now, there are no known archaeal pathogens. You have often heard that these organisms tend to inhabit extreme environments, so high salt environments, high temperatures, but it's not a rule. As we work through this section on archaea, we're going to find out that their metabolism is a lot like bacteria. They do things with carbon, for the most part, that look like bacteria. However, their informational structures, their ability to replicate DNA, when we transcribe DNA into RNA, when we translate, those pathways and those proteins that drive those pathways look far more eukaryotic than they do bacterial. So for a while, researchers have suggested that the archaea were sort of like a stopping point, as during the evolution to eukaryotes, right? That is, did bacteria evolve into archaea that evolved into eukaryotes? Now, it turns out that there are some people that actually do believe that, which is sort of different than the three domain, that is, bacteria, archaea, and eukaryotes have all evolved separately. And I'll show you a paper a little bit later on in our discussion that produces some very interesting data that suggests that the eukaryotes actually did evolve out of one group of the archaea. I don't know how many people actually really believe that, but there is some very tempting evidence out there. I don't think I want to move on anymore. I'm going to get you guys out of here. I will see you on Wednesday. We'll see you on Friday.
That's the question. That was probably very